Hello everyone, this is Jimin Guo from the University of New Mexico. Today I will talking about my poster, Biomimic Rebuild of Multifunctional red blood cells. So as we know, red blood cell is the simplest cell in structure, lacking a nucleus and most intracellular organelles, providing a suitable target for the rebuilding and biomimicking the multifunctional artificial cells. So red blood cell possess 4 different main characteristics. The first is the specific shape, and then the delivery oxygen from the lung to the bodies, and then the flexibility and the lung circulation properties. In this study, red blood cell mimic particle will be constructed using 3 separate building blocks. The silica, bioreplica, and layer by layer self-assembling process will transmit red blood cell into the flexible red blood cell shaped polymer core, which maintain their specific shape and the flexibility. Then the loading functional cargoes will provide specific cargoes after encapsulated with the red blood cell native membrane, will mimic the native red blood cell membrane surface properties to achieve lung circulation properties. From this image, we can easily find the red blood cell, rebuild red blood cells can have the same as the native red blood cells that bioconcurrent shapes. As we know, the flexibility is another key property of enable red blood cell to easily trans the microvasculature with the dimensions smaller than their size and display a long circulation time in vivo. So for this case, we're building a microfluidic blood capillary models, which is similar as the in vivo that are microvasculature systems. Then we say the red blood cell mimic particles with 4% of the cross-link concentration behavior totally the same as the native red blood cells. This is suggesting our rebuilt red blood cells might show the similar in vivo circulation properties as native red blood cells. To further emphasize this, we inject our rebuilt red blood cells inside the chicken embryo models and the mice models. So as we see, it has circulation very well inside the chicken embryo vasculature. And for the mice models, circulation half-life is close to 2 days, which indicate a long circulation time. It is also learned that red blood cells could help oxygen transfer from the lung to the body tissues. So after loading, the hemoglobin rebuilt red blood cell showed a similar that oxygen curve as native red blood cells, suggesting the hemoglobin loading the rebuilt red blood cells can be a possibility substitute for the native red blood cells. Additionally, different cargoes such as TPPS, DOX, oxidized, ioxidized nanoparticle, and biosensors could be loading onto the red blood cell mimic particle to provide a broad range of the potential applications such as MRI, contrast, anti-cancer, and magnetic properties. So that is what we do here. In the future study, we will focus on the biomedical applications of rebuilt red blood cells, such as the clearance of the blood toxin or the toxic metabolic inside the blood, or targeting delivery for the liver using rebuilt red blood cell, and improve the short half-life components in the circulation. That's all. Thanks for your attention.